Last video said there are many different types of proteins. We are going to see that some of the different types of proteins include the marker proteins. Here's a lovely example. It has a carbohydrate chain attached. We are going to see that there are receptor proteins. A receptor protein has a hole at the top that is going to allow for a substance to attach to it. We have transport proteins that either create a nice channel or are going to be able to pump different molecules through across the membrane. We also are going to see that there are enzymes that are involved and that there are anchoring proteins. These curly Q's are different types of proteins, and they are anchoring into the cytoskeleton. The major purpose of a marker protein is to identify the different type of cell or let other cells know what it is. It does this with a carbohydrate attached to the outside. So this carbohydrate that we just showed you, this carbohydrate that's right here, is going to essentially let other cells know what kind of cell this one is. Because this protein, the protein is this purple portion, has this carbohydrate attached, we can call it a glycoprotein. We also are going to see that there are such things as receptor proteins. As I said, a receptor protein essentially has this little tiny space at the top that's going to fit directly with some type of substance. When that substance comes and fits into this portion of the protein, this will change the shape of the protein. And when the protein shape is changed, it will send a signal inside of the cell. That will allow the cell to know what's going on in its surroundings and what it needs to be doing in order to adapt. A lot of times these are going to be how hormones work, where a hormone will come and fit into that spot exactly, and then it will change the shape of the protein and the cell will receive a signal from the rest of the body. The next major type of protein are transport proteins. They can help with either passive diffusion or active transport. In passive diffusion, we're still going from a high concentration to low concentration. Kind of like what we've been talking about with all sorts of diffusion. Think about the perfume. This is simply going to be a channel. It creates a larger hole for large molecules to pass through the membrane. A molecule like glucose is too big to simply go across the phospholipid bilayer. So instead there's a channel that creates a hole and the glucose can pass through. Active transport is a little bit different. It goes against the concentration gradient, meaning that the molecules are going to go from a low concentration to a high concentration. And this is going to require energy. So we're going to see that the protein literally pumps the ions or whatever needs to get across through the membrane using energy. If we take a look at a picture of this, we see here that in this diagram that we have a high concentration of green particles outside of the cell and a low one inside. The particles could go through just the phospholipid bilayer, no protein involved. However, they would have to be small in order to do that. If they are too large to do that, if there was a channel protein, like this one right here, it would be able to go through that hole that the protein creates and go across the membrane, going from high to low, no energy required. If, however, it wanted to go from low to high, go back against the gradient, then we would need to add energy. This next diagram gives us a better idea of how a protein pump works. Outside of the cell, we have a high concentration of sodium and a low concentration of potassium. We want to pump the sodium out of the cell. So the sodium binds to the actual spot of the protein and energy comes in and attaches. As the energy gets used up, the protein changes shape and the sodium is released outwards. Same thing happens with the potassium. Potassium comes in, attaches, the energy gets used up, the protein changes shape and moves across the gradient or against the gradient to